The next speaker is walking over to the podium, Bob Casper. Bob Casper is the chair of translational medicine at Mount Sinai Hospital in Toronto. And he's going to talk to us about the clinical application of oocyte supplementation by autologous egg precursors, mitochondrial transfer.
Thank you, Dominic. Good morning, everybody. I also want to thank the organizers for the invitation today. And I want to thank the other speakers because they've essentially given my talk for me. So I have the luxury this morning of focusing on some of the areas that are more controversial in a review of mitochondria, and to then go on and show you our clinical data with autologous mitochondrial transfer, and some very interesting new data on frozen and fresh mitochondria for injection that I think speaks to the biologic plausibility of this treatment. These are my disclosures. And I just want to start off by saying what we all know, that IVF has been around for a long time. We have good success rates presently in good prognosis patients, but the ones who fail repeatedly to get pregnant with IVF or have poor embryo development, our options are really quite limited today. It's thought, as the previous speakers have told you, that energy in the form of ATP produced by mitochondria is important for embryo development. And as you've also heard, the egg mitochondria are different from our typical picture of a somatic cell mitochondria shown on this slide. The egg mitochondria are morphologically round. They have a very dense membrane and sometimes very dense matrix. There also seem to be a lot of them. So this is an oocyte, a human metaphase II oocyte stained with JC1 dye that you've heard about that looks at mitochondrial membrane potential. The green dye is showing you mitochondria with low membrane potential, so these are thought to be relatively inactive. And you can see that most of the active mitochondria around the outside of this egg, they're the red ones, and they're probably at this stage participating in cortical granule reaction. You already heard that there are 200 or more mitochondrial DNA copies in a mature egg. And this is the controversial area now, because it was presumed in the past that there was a one-to-one relationship between the number of mitochondrial DNA copies and the number of mitochondria. This now looks like that may not be true. Jonathan van Blerkum has done a very sophisticated study where he actually counted the number of mitochondria in a blastocyst and then extrapolated back to the oocyte. And his calculation was that there was probably between 25 and 30,000 mitochondria in a mature oocyte, not 200 or 300,000. And I think that's probably correct, because if you remember Justin's graph of how rapidly mitochondrial DNA copy number increases during oocyte development, I think that's much too rapid for actual new mitochondria to be formed or to replicate. It makes much more sense that this is replication of mitochondrial DNA instead. So we're probably talking nowadays in a mature egg like this around maybe 30,000 copies of actual mitochondria. But there's still a lot more mitochondria, or certainly a lot more mitochondrial DNA copies than in somatic cells. And again, I think Justin went through this with you earlier, that the reason that there are so many copies is that the mitochondria get diluted as the embryo divides multiple times, so that by the time you get to day 5, where you have about 100 cells, you're probably back to the same number of mitochondria that would be in most somatic cells. And that's also why most of the mitochondria that we see in that mature egg are green, They're actually turned off. They're waiting to be switched on when they're needed. So how can we enhance mitochondrial activity? We've done some work looking at vitamins, nutritional supplements, but I think one of the first studies was the one by Jacques Cohen. Again, I think this was mentioned earlier. And the idea is to transfer cytoplasm from a young, healthy egg into an egg that has, or into eggs from patients who have suffered repeated poor embryo development, with the idea that that cytoplasm may facilitate embryo development. And the idea actually came from the mouse, because there are mouse strains that have a 2-cell block and other mice strains that don't have a 2-cell block in vitro. And if you take cytoplasm from a mouse that doesn't have a block and inject it into a mouse that has a block, you actually overcome the block. So I think that was sort of the initial place where the idea came from. In any event, this procedure was successful. There were many babies born worldwide. And this slide just shows you in detail exactly what's involved. So it's really an ICSI procedure where a small volume of donor oocyte cytoplasm is aspirated together with the sperm, and then they're both injected into the oocyte. And you can see here that this is sort of the volume of cytoplasm that's injected, and here this is outlined with the computer. So you can see that it's probably relates to a volume of around 5 picoliters together with the sperm. The problem, though, as has been alluded to, is that many of these babies were shown to have heteroplasmy, that is, 2 different populations of mitochondrial DNA. And at the present time, the long-term effects of neutral mitochondrial heteroplasmy, that means that although there are 2 different types of mitochondria, they're both normal. Neutral mitochondrial heteroplasmy, we really don't know the long-term implications of that. And as a result, ooplasm transfer is currently sanctioned, I think, worldwide. And certainly in North America, it's not allowed to change the genome of the embryo that could be transmitted to subsequent generations. So certainly mitochondria would fit into that sanction. So the new option that I want to tell you about today, and what we have some experience with, is autologous mitochondrial transfer, which may be a new option for repeated IVF failure. So the goals of this treatment are to take the patient's own mitochondria, inject them into the eggs with the hope that we would be able to improve oocyte and embryo energy, which would result in better embryo development and successful live births. And as Evelyn Telfer just told you, there's been lots of controversy in this area, but I think there are many studies now suggesting that egg precursor cells do exist. They seem to be in a dormant or quiescent state in the cortex. They are unipotent. These are not stem cells. They are germline cells that seem to be able to form oocytes. And because they're sort of sitting there dormant, the thinking is that they may be more immature, younger, healthier, and have more healthy mitochondria than the aged oocytes. And morphologically, the mitochondria from these egg precursor cells look very similar to oocyte mitochondria. So here's a mitochondrion from a brain cell. You can see this elongated morphology with multiple cristae. Here are mature human oocyte mitochondria, and here is an egg precursor cell. This is the nucleus, and here you can see the mitochondria inside this cell, which they're round. They look very much like the mature egg mitochondria. So the advantage of injecting these mitochondria is that, first of all, we're not changing the genetic makeup of the embryo or the egg. We're putting the patient's own mitochondria in. So we essentially maintain what's called mitochondrial homoplasmy. And there is communication between the nucleus and the mitochondrial genome. And that should be maintained because these should act like other oocyte mitochondria. So just to sort of give you an idea of what's involved clinically, first of all, we have to get some ovarian tissue in order to get these oocyte precursor cells isolated. So this involves a laparoscopy and ovarian tissue biopsy. This is just done with scissors. It's a very superficial biopsy. The piece of tissue is about 6 millimeters by 18 millimeters in size, and it can be, doesn't have to be all in one strip. It can be multiple biopsies. That tissue is frozen on site. It's then transported to the Ovascience lab in our clinic. And over the next 2 or 3 weeks, that piece of tissue is thawed, it's digested, and the oocyte precursor cells are identified using flow cytometry and this DDX4 antibody that Evelyn told you about. The number of egg precursor cells is counted, and then they're frozen again. And for us to proceed with this procedure, we'd like to see between 50 and 70,000 of these cells. The lab then gives us the green light to go ahead with an IVF cycle. So about a month after the biopsy, the patient undergoes standard controlled ovarian hyperstimulation. We do an oocyte retrieval. And on the day of the oocyte retrieval, those egg precursor cells that were frozen are thawed. The membrane's disrupted, the mitochondria are concentrated using differential centrifugation, and then the egg precursor cell mitochondria are injected into the egg at the time of ICSI, and then we look for embryo transfer. And we grow these embryos to day 5. That's the aim, is to take the embryos to day 5 before transfer. And the patients that we're selecting for this process are patients who've failed IVF in the past, mainly because they're not getting their embryos to day 5. They're resting between day 3 and day 5. So I want to show you some of our clinical experience with this autologous mitochondrial injection. So you can see that we've broken things down into age. So there are patients below the age of 40 and patients above the age of 40. So we started here with 26 patients who had a mean age of 35 years. You can see they had an average of 2 failed previous IVF cycles before the augment treatment. And all 26 of them had the biopsy and the autologous mitochondrial transfer at the time of ICSI. And we did get to blastocysts in this group. So we had 18 fresh plus 18 frozen embryo transfers. The ongoing pregnancy rate per fresh embryo transfer was 17%. Interestingly, the ongoing pregnancy rate per frozen embryo transfer was 61%, which gives us an ongoing clinical or live birth rate per cycle initiated of 54%, or per embryo transfer of 39%. And we now have 12 live births. These babies are born, and they're all healthy. It's a different story completely in the patients over 40. So we had 23 patients, mean age 42.5 years. They had a previous history of, on average, 3 failed IVF cycles. We did get blastocyst development, 15 fresh transfers and 14 frozen transfers. But in contrast to the younger patients, we had no ongoing pregnancies with fresh transfer, although there were 4 spontaneous abortions early on. And of the frozen embryo transfers, we had 1 pregnancy out of 14, plus 2 spontaneous abortions. So our pregnancy rate, ongoing clinical pregnancy rate per cycle start is around 4% in this group. Now, since the beginning of 2016, Ovascience has introduced the use of frozen mitochondria at our clinic rather than fresh. And I think the advantages are obvious, but if you have frozen mitochondria, this allows you to ship them outside of the main clinic, which means that we can decentralize this procedure. So other labs could do the ovarian biopsy. It's just standard freezing technology. That frozen tissue can be sent to the Ovascience lab, the mitochondrial preparation is produced, and then the mitochondria is sent back to the original lab, and they can then do the autologous mitochondrial injection together with the ICSI. So I think that will really facilitate the development of this procedure. And Oviscience has done several studies looking at the fresh and frozen mitochondria. Here you can see an egg precursor cell. In this one field, you can see maybe about 15 of these of the mitochondria in that small cell. And then this is the morphologic appearance of the concentrated mitochondria before freezing, and this is after freezing. And you can see they really look morphologically the same. One of the tests that is important to know is whether the cell membrane of the mitochondria is leaky or not, whether they're leaking out cytochrome C. And what we found is that using different cryoprotectant protocols, with at least 2 of them, we're seeing very little, if any, cytochrome C release, suggesting that these mitochondria are intact post-freezing, either using slow freezing or vitrification. And if you just look at ATP production from the frozen mitochondria, the patterns you can see here are very similar to ATP production in fresh mitochondria, either with a TCA cycle substrate, succinate as a substrate for ATP, or without. And both graphs look very similar. So this is, I think, is really now getting into some interesting data. So this is showing the number of eggs injected with fresh mitochondria, so it's 61, versus frozen mitochondria, the fertilization rate, and the blastocyst formation rate. So this is fresh or frozen in women under 40, and this is fresh and frozen mitochondrial injections in women over 40. So first of all, just look at the fertilization rate. So this is ICSI. The fertilization rate is running around 50 to 60%. Normally, in our lab, we're running about 75 to 80% fertilization rate with ICSI. So this really, I think, speaks to Justin St. John's point that if there's a mitochondrial deficit already in these oocytes, that the fertilization rate is going to be lower. So this data is very supportive that we're dealing with poor prognosis patients who have a low fertilization rate and probably low mitochondrial DNA copy number to start with. The second thing to look at is that there's a very good blastocyst development rate. So we have, with fresh mito injection in the younger patients, we have a 47% blastocyst rate from the eggs that fertilized. And with frozen mitochondrial injections, it's about the same, 44%. So I think that's a really excellent blastocyst rate that we would expect to get in younger patients who are healthy. In the older patients over 40, we had a 31% blastocyst rate with fresh mitochondria and about 42% with frozen. So really, I think if you look at these two together, the blastocyst formation rate is pretty much the same as with the younger patients. And again, this sort of tells us that likely this treatment is working. We're actually increasing the energy. We're getting more embryos to blastocyst. The reason they're not implanting, though, in this older group is because we can't change the chromosomal makeup of the egg by injecting more energy. So I think if we're dealing with older patients, especially ones with repeated failure, who probably have a lot of aneuploidy in their oocytes, injecting more energy is not really going to be helpful. So in conclusion, it looks like autologous mitochondrial injection during ICSI has some promise for improving pregnancy rates. It's actually going to be valuable, I think, for a very small niche of patients, because we're really talking about patients under the age of 40 who have pretty good ovarian response, but whose embryos arrest between day 3 and day 5. So we're probably talking about a very small population for which this would be warranted. And I just want to end by thanking our TRIO clinic, especially the TRIO lab, who've been responsible for all the injections. Andrea Jersakova, who's been my long-term research associate at the Lunenfeld Tannenbaum Research Institute, and Oviscience for for providing some of the data that I showed you today. Thanks very much, everybody.